All right. Okay, well, we're going to continue to take a look at RNA interference. Now, we went through the process and stuff, and the idea, once again, you get these small microRNA genes that get processed in the nucleus, cut into a small double-stranded, what we call small interfering RNA in the nucleus by this enzyme called dicer. That's the one with the axe. And then it goes in the cytoplasm, binds that enzyme complex called the RISC complex, RNA-induced silencing complex, which contains an enzyme that digests RNA. Those small interfering RNAs then will bind to complementary sections of messenger RNAs, often located in the 3' untranslated region. It can go to coding region too. And when that happens, that's either going to stop translation and slowly send these guys to the RNA degradation sites, the so-called P-bodies, or if you have a complete match, it activates the enzyme in the RISC complex, which destroys both of the RNAs. So either way, you end up shutting off that RNA. It can't be used anymore. So that's another step of regulating at the level of translation. And clearly it's very important because if you get rid of the genes for some of the enzymes involved in this, you get all kinds of gross abnormalities in development in both plants and animals. Things overgrow, other things undergrow. Yes?
Okay, the mRNA is going to bind to the siRNA at the 3' region?
Well, many times the complementary regions in the 3' untranslated region of the messenger RNA, but not always. There's no reason you can't go after something in the coding region too. Okay, well that's the basic thing. Clearly it's developmentally important, but but it's also a very powerful research tool and a potential clinical tool. Because what we can do is, and there's companies that will do this. In fact, I think one is based in Huntsville. What you can do is make an artificial small interfering RNAs. You can buy these things. You tell them a sequence. Let's suppose you have found a gene. You get the sequence of that messenger RNA, and you look for a unique feature in that sequence that you don't find elsewhere in any of the databases that have these sequences. You find a unique feature, a unique sequence, and then call these guys up and say, hey, I want an siRNA that will target this sequence. And then you fork over some federal grant dollars. It's actually not that much, it's several. I think they do them for several $100 now. And they will send you back a week or so later this artificial siRNA, which then you can introduce into cells. And the idea behind this is that will activate the RNA interference mechanism and effectively ramp down or shut off the expression of that particular gene. Okay?
Apparently they would love this Mario.
What's up?
Folks in the hall.
The people in the hall that are talking very loudly.
Oh,
so
okay.
that's
All right, okay. Okay, so reduces the expression of the targeted gene, often over 90%. Now, generally RNA interference in this method does not shut the expression of the gene off entirely. There's usually a little bit of expression left, okay? But it ramps it down a great deal. So they often call this technique a gene knockdown rather than knockout, because it doesn't completely shut off expression of that gene, but it knocks it down enough that generally you're going to see some kind of phenotype, some kind of effect. If you knocked it out 90–95%, chances are you're going to get some kind of effect, even though there's a little bit of gene expression left. Now the advantage of this is the old way of doing what's called a gene knockout, that I described that as a technique called homologous recombination. I described that in the introduction. Don't worry, you're not going to have that in the test, by the way. But anyway, that technique, although it effectively disrupts and is used to completely disrupt the gene and you have no expression at all, that technique is extremely arduous. You can often spend a year or two just trying to get this done. These gene knockdowns are a lot easier and quicker to do. You could do an experiment in a few weeks. So, it allows you to start looking. You find a new gene, you ask yourself, what the heck does this gene do? Well, you can use RNA interference to get some insights in what that gene does by shutting off or shutting down its expression and seeing what happens. So a lot of times, and people have done this, using these techniques, for instance, the first eukaryote they had the entire genome sequence was yeast. It has about 6,000 genes in there. Half of them at the time had no similarities with anything in the database. They had no idea what these things were. So a group of labs got together and said, hey, let's shut off the expression of each and every one of those genes in different strains of yeast and see what happens. It took them a few years, but they got it. They know the function of every single one of those 6,000 genes. And they're doing that now with other organisms, including that little roundworm C. elegans that was the first multicellular eukaryote. So here you can see, what does this gene do to develop? What happens if we shut it off or shut it down quite a bit? So it's a powerful technique and it's a lot easier than the old style way. So it's a very big research tool. It's a hot topic and a widely used technique nowadays in molecular biology. No, we don't do it at JSU. That takes a little bit more money than what we often get. But it's a powerful molecular biology technique to investigate what a particular gene does, what its function is. Look like you have a question.
Well, so they use that type of they use this process to map out every gene's function?
Yeah, not all in the same yeast strain, but you have one strain of yeast and say, okay, we're going to shut down gene X. This other culture of yeast, we're going to shut down gene Y, and all the way up to all the 6,000 or so these guys got. So you can see the effects of each and every single gene in different yeast. Do they behave normally? You know, is their metabolism normal? Do they divide normally and things like that? Yeah. So it's still a lot of work, but to investigate the function of thousands of genes in only a matter of years, and there was a whole bunch of labs that did it, that's quite impressive. Okay, so that's one kind of thing. A research tool. It's a powerful research tool. And there are potential clinical applications. Okay, there's nothing with RNA interference out in the doctor's office yet. There is at least one RNA interference-based drug in human clinical trials. They just started the clinical trials of this. There was a lot of research and development by the different pharmaceutical companies, but various hitches and stuff. It wasn't something that looked like it was going to make a short-term profit for them. And of course, like all the countries in the U.S., they're always looking for the short-term quarterly report, not where they're going to be 10 years from now. So they kind of cut back at some of the research. But there is something that's in clinical application, that's in early-stage human clinical trials at this point. And the idea is this. If we can make, let's suppose we have some kind of disease. In fact, cancers are a perfectly good example. Many cancers are caused by overexpression of certain genes in certain cell types. Now, if you can deliver artificial siRNAs and get them into those cell types, you can ramp the expression of that gene down. And that could, for instance, slow down the growth rate of cancer cells. That's the idea. Another example, flu. You know, you go out and get a flu vaccine, and it's specific for one particular strain. Matter of fact, the flu floating around, even the vaccinated people, there's a second strain around that's widespread. And even the people that are vaccinated against the first strain have very little defense against the second strain, so they get nice and sick. Okay, well, if you can use an RNA interference-based method, that problem with vaccines is they're going to use your immune system, which targets the surface proteins on the flu virus. And those are designed to mutate rapidly. That's why you get so many different strains, and that means the virus stays one step ahead of the host's immune system. But inside there are certain genes and certain proteins that are absolutely essential for viral function and viral replication. If you could ramp down the expression of those genes, you could have a drug that was independent of strength. It would shut off or shut down the expression of any flu virus. There's a lot of potential in that. Now, so far, like I say, nothing in the doctor's office yet, but it's been investigated for things like certain kinds of viruses, flu, HIV's another one, various kinds of cancers that are due to overexpression or misexpression of this gene or that gene or what have you. So there's a lot of potential, but it hasn't been realized yet. Maybe 10 or 20 years from now, there'll be a number of RNA interference-based therapies right in your doctor's office. So that's the hope for this. So clearly, RNA interference is a powerful technique. Now, there is one little potential thing you have to worry about with this. You have to make absolutely sure, if you're going to make an artificial small interfering RNA, you have to make absolutely certain that you're targeting one gene and one gene only. Recall we said that RNA interference, a given small interfering RNA, may regulate the expression of numerous genes because it goes after sequences that are common to all, especially ones that target the untranslated regions. Those are a little bit less diverse than the coding regions. Okay, well, case in point, some years ago, a strain of mice, these investigators wanted to shut off the expression of a particular gene that had something to do with the immune system. So they went out and ordered some small interfering RNAs, got them into these mice, introduced them in these mice, and within a day or two, every single one of them died. What happened there? Apparently, there was some other sequence. That RNA, that small interfering RNA, targeted a sequence that was found in addition to a gene that's expression was absolutely critical for life. And when they shut both off, and of course, end result is they lost their whole mouse colony, at least the experimental samples. So there is a risk that you have to make absolutely certain if you're going to use this in the doctor's office that you're going after this gene and this gene only, and it's not going to affect the expression of any other gene, because otherwise you can get some extremely unpleasant surprises. Okay, so that has a lot of potential, like you say, both as a research tool and as a potential clinical tool, although the clinical applications are still in the future. When this first came out, it was extremely hot. It still is a very hot topic, but it's being widely used now. Okay, yes?
I was watching House this weekend,
Uh-huh.
and on the episode that I was watching, they said that they were gonna do a clinical trial on this person, and it was this that you're talking about. And I was like, yes, I know what that is.
Yeah, you're probably out half a million, out of all the people in Alabama who are seeing this, you're probably one of a handful who know what that is. Well, really,
I felt so smart.
let's face it, there's an awful lot of people in this state that are largely literate and have barely gotten out of high school. You know, go down to Walmart, you see a lot of them. No, well, not here, not so much here, we get a lot of students there, but you go to some of the Walmarts around and you see these people that just look so sad because, you know, here they are getting exploited and that's the best they could possibly do. But yeah.
Well, thanks to you, I understand.
Okay, all right. Well, like I say, that actually, there is a clinical trial for at least one of them going on now. It's in the early stages, even if everything works, and I forgot what was going after. But even if everything works as advertised, it'll still take several years to get it into the doctor's office. It takes a while. You have to do like 3 or 4 stages of clinical trials. The first one is to see whether it's safe and, you know, it's not going to kill the patients or something. And then the next step is to get an idea of the dose and the duration and all that kind of stuff. And then you start doing a larger sample with several hundred, maybe even 1,000 people or so to see if it's safe and effective. And you go give all this data to the FDA and they say yes or no. Once in a great while for some things, political things get into the way too, but that's generally not the case for a lot of things. But anyway, and if it's something that looks extremely promising, there's sort of a fast track approach where they can get things in real fast. If it has like a dramatic effect, you know, you find this siRNA, you know, gives an 80% remission rate for lung cancer or something like that, it's gonna get fast tracked. Yeah, Okay, all right, so powerful research and powerful clinical, and or at least potentially powerful clinical too. But there are other kinds of RNAs that play other kinds of regulatory roles. We're just going to touch on one or two of them in a moment. But